Continuing on with the test review. For test 1, after the tertiary level of structure of the protein, where it becomes functional, its functional shape, some proteins enter the 4th level of structure quaternary, where you'll have like 2 or 3 or even 4 of the tertiaries come together. Best example of that, I didn't mention it in class, though, is hemoglobin. We talked about this in AAT2. Hemoglobin is a very important protein for transporting oxygen in the blood and in red blood cells, and it's an example of a protein that has to get to that 4th level of structure to be functional. Okey-dokey. All right, also, besides knowing the levels of structure, make sure you know about enzymes. Many enzymes are proteins, and enzymes work as catalysts. They speed up chemical reactions. They speed up chemical reactions, and they do that by lowering the activation energy. NRG. It's the energy that's necessary to start a chemical reaction. So they lower how much energy is necessary to start the reaction, and reactions can proceed faster. You have to worry also about denaturing proteins and enzymes. Denaturing them can make them non-functional. Remember, there are 3 ways to denature: change the pH, the temperature, or the salt concentration. Those are 3 major ways to denature proteins and or enzymes. All right. Any questions? I hope you liked this review, though. Snapshot. Did it all already. All right, last would be nucleic acids, DNA and RNA. Nucleic acids, the building blocks, are called nucleotides. Who just came in? Was that another one of my kids? I only have one, that was a joke. Nucleotides are the building blocks, and they have 3 parts to them. They have a pentose sugar, or carbohydrate, so just write pentose. That means 5 carbon. 5 carbon sugar, a nitrogenous base, that means they have nitrogen, and a phosphate group. And those are the 3 parts that make up a nucleotide, which is the building block. Okay? And then really quick, the difference between DNA and RNA. You've already written this down, but DNA is double-stranded, usually. Remains in the nucleus, and they have different sugars. They also have different sugars and different bases, nitrogenous bases. And RNA is usually single-stranded. It can go outside the nucleus. It travels throughout the cell. And there's different sugars and bases. Plus E. Okay, ATP is a variation of nucleic acids of a nucleotide. It's the energy. I should say the major, because it's not the only one. I don't want to tell you a non-truth. It's the major energy molecule of the cell. It has high energy phosphate bonds. questions? All right. Chapter 3. We have the fluid mosaic model, which is what makes up the cell membrane. You should have an idea of what makes up the cell membrane. Fluid mosaic model means that the cell membrane is constantly changing. It's in a liquid state. So it's basically a phospholipid bilayer. So it's a layer of phospholipids like this. Right? With a randomly embedded cholesterol molecule, which is there to prevent the phospholipids from packing tightly together and help keep it in the fluid state. There are proteins. The integral proteins span the whole entire phospholipid bilayer, and the peripheral proteins are just on one side or the other. So you have integral and peripheral proteins. Integral proteins are usually channels or carriers to get things across, and peripheral proteins are usually enzymes or receptors. Also on the outside of the cell, oh my gosh, let me draw this quickly, you would have carbohydrates attached to the proteins or to the lipids. All those carbohydrates are called the glycocalyx. I don't know how to spell it. But there's too many vowels or consonants at the end. The glycocalyx is necessary for cell-to-cell recognition. It helps us recognize, our bodies recognize one cell from another. Cell-to-cell recognition. All the carbohydrates on the outside of the cell. Would be the glycocalyx. All right, just skipping to movement, passive and active processes, movement across the cell membrane. So we have passive movements are anything that is a type of diffusion, whether it is simple diffusion, carrier-mediated diffusion, channel-mediated diffusion, facilitated diffusion. Diffusion means that it's passive, you need no ATP, no energy is added. You're moving from a high concentration to a low concentration. Another way to say this is the molecule is moving down its concentration gradient. Down the gradient. That means going from high to low. Okay. So what can easily move across the cell membrane via passive diffusion would be something that is lipid soluble. Lipid soluble things move the easiest. Especially small and lipid soluble, because smaller things move more readily than larger. Small lipid soluble moves the easiest. The things that are not lipid soluble can also get across. They're just going to need the help of carriers. They can still go passively. So that would be either through a carrier, let's say that would be for a carbohydrate, like glucose, or an amino acid. And then through a channel, usually through channels, our ions go through. And then of course we have osmosis, which is the diffusion of water. You can only use osmosis for the word water. And water can go across the cell membrane or via just simple diffusion, or else use an aquaphor or a channel. Neurotic. Not as happy as I would be if the pen worked. That would be You're going to need to add ATP. You move from a low concentration to a high concentration. And the best example is the sodium potassium pump, where it moves 3 sodium out of the cell, and then 2 potassium into the cell, and uses 1 ATP. I don't know if I'm going to get through everything, FYI. Also, while I'm here, let me talk about hypo, iso, and hypertonic solutions. But are we okay with the active transport, though? I mean, that's like a quick review on that. There's more to it. You're going to have to look at that yourself. Hypotonic means there would be less particles, more water outside of the cell. Iso, same particles, same water. Hyper, there would be more particles, less water outside of the cell. Put a cell in a hypotonic solution and it will swell, and it could even burst. Isotonic solution, it stays the same, and hypertonic solution, the cell will shrink. And remember, the particles do not move across the cell membrane, it's the water that moves. Any questions on that? So, exocytosis, things are leaving the cell. Endocytosis coming in, those are types of active transporters on your list. The resting membrane potential is the charge across the cell. When the cell is at rest, and when the cell is at rest, it's negative, more negative inside compared to outside. It's actually, the value is about minus 70 millivolts, more negative inside than outside. Okay? That's the resting membrane potential. And when you talk about ions moving in or out of the cell, you've got to worry about their concentration gradient and the charge gradient. That's called the electrochemical gradient. And you need to think about that when ions move across. The types of junctions, we have a tight junction, which is a permeability barrier. We have gap junctions, which are channels for cells to communicate with each other, like sending chemicals across from one cell to another. And then we have desmosomes. They attach cells to one another. I think what I have left now are the organelles and their functions. I think that's about it, right? The very last thing. I feel like I just have to do it really quickly, like in 2 minutes, okay, 2 seconds. So the nucleus houses the DNA. That's where the DNA is located. Okay? Mitochondria, that makes ATP. Ribosomes make proteins. The proteins can then go to the rough endoplasmic reticulum, which will process those proteins. Then those proteins could go to the Golgi, where the proteins are further processed, stored, packaged to get ready for secretion. Then you would have a secretory vesicle that brings the proteins to the cell membrane. For secretion out of the cell. A lot that I just wrote there. You have the smooth endoplasmic reticulum that makes carbohydrates, lipids, and can store calcium. Ca+2 is calcium. Lysosomes contain hydrolytic enzymes to break things down. Perioxysomes are for detoxification. Centrioles for cell division. Probably did everything, right? You feel that? What was that? Lysosomes? Hydrolytic enzymes, perioxysomes, detoxification, centrioles, cell division. Is that, did I answer the question?
No, it's just gonna be
Cytosol is the area between the nucleus and the cell membrane. And cytoplasm is the gel-like stuff in the cytosol. But the cytosol and the cytoplasm are not organelles. The cytosol is an area and the cytoplasm is this gel. Organelles are the ones above that. Okay. That was a quick and dirty review. Remember, it is a review. It is not substitute reading the book or a lecture. Thank you for joining us.